a signal molecule binds to the G protein linked receptor. This activates the previously inactive G protein by displacing the GDP with a GTP molecule. It then binds to adenylyl cyclase, making it active. This initiates the signal transduction pathway. ATP binds to the enzyme and gets converted to CAMP. This CAMP molecule allows for the amplification of the signal, triggering the phosphorylation cascade. The phosphate is then released from CAMP and attaches to the protein kinase. As more phosphates are released, it begins to move down to the endoplasmic reticulum, releasing the calcium ions into the cell. With type 2 diabetes, the initial signal molecule, known as insulin, does not bind to the G protein linked receptor. Without a signal molecule, the G protein remains inactive and the cell cannot produce a response. Symptoms include increased thirst and urination, increased hunger, fatigue, blurred vision, numbness or tingling in the hands or feet, sores that don't heal, and unexplained weight loss. Type 2 diabetes can sometimes be treated by simple lifestyle changes. For example, diet and exercise can often improve blood glucose levels. It can also be treated with medications and insulin injections. With the tyrosine kinase system, two signal molecules must bind to the receptor tyrosine kinase proteins. These proteins are then activated and they bind to each other, forming a dimer. ATPs drop off one phosphate which bond to the tyrosines. These 6 ATPs turn into 6 ADPs and exit the system. The inactive relay proteins attach to the phosphates, which activates them. These activated proteins carry the phosphate and bind to the protein kinase. This then activates the protein kinase, and as more phosphates are released, they begin to reach the ion-gated chain. These phosphates open the chain, which releases the calcium ions.